Hi ladies, so I'm going to try and talk you through some of the kind of initial stages of genetics. Okay, so that's looking at DNA and RNA. Okay, so let's start off with DNA. Now you need to know what DNA stands for first of all. So it's a lovely Scrabble name, okay, good one for Scrabble. Deoxyribonucleic acid. Deoxyribonucleic acid. DNA, girls, is found in the nucleus. There's also some small amounts found in the mitochondria as well, and chloroplasts in plants. Okay, so there's very small amounts there. Its structure now, girls, usually DNA plus histones. There's proteins that they're wrapped around, and they help the DNA fit into the nucleus into a really tightly coiled structure, okay? And that's what chromosomes are. So chromosomes are made up of protein and DNA tightly coiled together. And the bases, girls, that they're made of has a structure of a double helix, the DNA itself. And that was discovered by Watson and Crick. Scientists, okay? Your bases, you've got adenine, thymine, cytosine, and guanine, okay, which you shouldn't know the full names of. These two guys are called, so A and T go together, okay, and G and C go together, all right, and they're paired together by hydrogen bonds, all right. So adenine and guanine are called purines, and thymine and cytosine are called pyrimidines, okay? Now A and T are bonded together by 2 hydrogen bonds. G and C are held together by 3 hydrogen bonds, okay? So that's what holds the steps of the ladder together when you're looking at a double helix, is those complementary base pairs, all right? So you've got your purines, your adenine, your guanine, your thymine, your pyrimidines is your thymine and cytosine, and you need to know how many hydrogen bonds hold them together. Now your nucleotide. So nucleotides, girls, are the basic building blocks of DNA. So that is your sorry, just change that there. So that is your sugar phosphate backbone. Okay, plus your base, your nitrogen containing base, okay? So your nucleotides are made of your sugar phosphate backbone plus your base there, and that's essentially the building block, the most basic kind of structure of DNA, all right? Now, let's look at a gene. So a gene is a set of bases that hold the information needed to make a protein. We'll talk about that in a second. So every trait that is controlled by our DNA, girls, it exerts its effect by the production of a certain type of protein, all right? Your eye colour, your hair colour, your skin colour, your freckles, whether you've got curly hair or not, okay, all of those traits are controlled by genes, which have their effect by making a protein. Girls, the genetic code then, okay, so that's also called a codon, all right? So every 3 bases code for an amino acid, and they combine in such a way, just like the letters of an alphabet, that they can make lots and lots of different types of proteins depending on the order that they're in, okay? So every 3 bases code for an amino acid. Now we also have non-coding, we also have non-coding DNA, or what's called junk DNA. I wouldn't say it's junk DNA because we still don't fully understand its structure or its function, but basically junk DNA is DNA does not control a trait, and it's not one that we fully understand what its function is for, okay? So there are some stretches of DNA that code for nothing and we don't really understand why. So that's junk DNA or non-coding DNA, okay? Now let's look at replication of DNA. So we've done mitosis, girls, and meiosis. So before either of those cell divisions have to take place, your DNA has to double in amount, okay, to ensure that the daughter cells either have the exact same amount of genetic information as the parent cell in mitosis's case, or have half the genetic information, a haploid cell for example, in meiosis case, okay? So during interphase, DNA replicates. Now the steps of replication are the following. So DNA unwinds. Remember, it's a double helix, so it becomes straight in structure, okay? It unzips. So those base pairs become broken, and that happens by restriction enzymes. Okay, then what happens, girls, is new nucleotides enter from cytoplasm, and they form a complementary complementary match to the unwound DNA. Okay, so complementary match to DNA, and then the DNA reforms. And it twists back in to the helix structure, okay? So this is really important, girls, to make sure that our amount of DNA in our cell doesn't change during cell division, because obviously we want to maintain that, because it's what makes us what determines what species we are. So hopefully that's clear. So DNA unwinds, it unzips by restriction enzyme, so that breaks all of the base pair hydrogen bonds, okay? New nucleotides enter from the cytoplasm, complementary match to the unwound DNA, so you end up getting a molecule of DNA which has one strand that was the old DNA and one strand that was the new DNA, okay? So you get twice as much DNA that you had there previously. It's called semi-conservative process because half the strand is old and half the strand is new, and then your DNA reforms into your double helix, okay? All right, so that's your replication of DNA. Now, before we go on to protein synthesis, girls, we're going to come on to RNA, because RNA is key to protein synthesis, all right? So your name, so ribonucleic acid. Okay, the types, we have a number of different types of RNA, girls. We have rRNA, which is ribosomal RNA. We have mRNA, which is messenger RNA. We have tRNA, which is transfer RNA, okay, or some people say travel RNA, but transfer RNA. Your structure, it's single-stranded, so it's not a double helix like DNA, okay? It has the base uracil instead of thymine. So in DNA we had A and T going together in C and G. So this time we have A and U going together in RNA rather than A and T, okay? So uracil is there instead of thymine, and it's found both in the nucleus, depending on the different types of RNA, and found in the cytoplasm as well, and also the ribosome. That's your RNA, ribosome. Okay, so that's your RNA. Now let's look at your protein synthesis, which I've partly rubbed out by accident. Protein synthesis. Okay, so protein synthesis. So you've got your initiation step, girls. This is where the DNA unwinds and unzips. Again, restriction enzymes. Okay, so your DNA unwinds and unzips, okay, so it's now ready for what happens next, which is your transcription, which is your mRNA enters from cytoplasm. And what your mRNA does, girls, essentially takes down the code that exists on your DNA. So it's recording the base pairs and their sequence, okay? So mRNA enters from the cytoplasm, okay? Then for translation, your mRNA leaves the nucleus and goes to the ribosome. Okay, and that's where your tRNA, your transfer RNA, bring the complementary amino acids to the ribosome, which then synthesizes your protein, which is going to have an effect, for example, your freckles or your hair color or your eye color. Okay, so let's go to that again. So initiation, DNA unwinds and unzips. Transcription, mRNA enters from the cytoplasm, okay, and will basically take the code that's there from the DNA. Translation, mRNA leaves the nucleus and goes to the ribosome. tRNA brings the complementary amino acids to the ribosome, and your protein is synthesized and therefore has its effect. So that's your protein synthesis. Now, let's have a little look at DNA profiling as well. So we did quite a few questions on this in class. So DNA profiling, girls, is used, for example, in crime scenes, also used to work out paternity of people, like who's the mommy and who's the daddy, okay? It also was used a few years ago when we had the horse meat scandal, where we had people selling horse meat for cow meat, so they were able to determine where the source of meat was, okay? So the steps of DNA profiling, girls, is DNA is released from the cells. It's cut into fragments, again using restriction enzymes. Separation by electrophoresis, which is running through a gel using electricity. And then we compare, we get these little patterns that look a bit like this, okay? That kind of look like bands, of coloured bands in gel, okay? And we compare them then, okay, to find out who the father was, or which DNA matches which suspect, okay? So what it's used for, girls, it's used for paternity, crime scenes as well. Also used in the food industry to determine the source of meat as well, okay? So girls, that is the first section of genetics, okay? I hope that's useful to you.